Hello everyone, welcome to this OncoDNA webinar. Today the topic will be the international status of next generation sequencing in oncology practice. My name is Aubrey and I will be your guide during this session. You are more than 200 registered people coming from all around the world, so I hope you will enjoy this session. All along the webinar, you will have the opportunity to raise your question by using the question box, just at the right side of your screen. We will take the time at the end of the webinar to answer your question. Now the time has come for me to present the two persons next to me, who are Jean-François Las, OncoDNA Chief Scientific Officer, and Marc Buchet, Marketing Manager. Guys, I give you the floor.
Thank you, Aubrey. So indeed, we have the pleasure today to present the result of the international survey we made about the status of next generation sequencing in oncology practice. So as you know, indeed, the importance of somatic molecular profiling of tumor, including NGS, has grown in the last decade. Due to the increasing number of clinical applications guiding the therapeutic choice of oncologists and also the speed of discovery of new molecular targets, needs are evolving very rapidly. So the objective of this webinar is to review the latest international NGS trends in terms of use technologies, panel size, interpretation platforms, and new requirements. We will show that the interpretation of molecular profiling for clear theranostic support for the oncologist requires now a deeper approach, deeper referencing, transparency, and reporting solution that only a few players like OncoKDM can provide. So how was this NGS Barometer done? So the international survey was done online. We sent a link by email via social professional network, pathology journal, and we sent them to thousands of pathologists, bioinformaticians, oncologists, and molecular biologists worldwide. It was conducted between the 14th of March and the 13th of April this year. So inclusion and exclusion criteria of this survey, we perform the analysis on participants using NGS or NGS data interpretation for oncology application and for somatic diagnostic application. We exclude participants not using NGS for oncology purpose. It was 20% of the participants, or using NGS only for germline oncology application. It was 15% of the participants. More than 300 persons participated to this barometer. As you can see on your screen, the C group, as illustrated on the breakdown chart, was used for the analysis, and this group represents 40% of the total of participants we had in this barometer. A first look at the origin of the participant. 48% of the qualified participants are located in Europe, while 21% are located in Asia and 13% in the Middle East. Regarding the specialty of the participant, molecular biologists and bioinformaticians represent 70% of the specialists, 47% and 23% respectively. We also asked the average number of samples they analyzed per year in the laboratories, and we reached an average on 750 samples per year. Half of the laboratory analyzed over 100 samples, while 16% analyzed over than 1,000 samples. The participants are working in private companies or in private laboratories, and 20% of them were in academic hospitals. So I invite you now to go through the result, and let's speak first about the type of panel, the panel size they use. So as you know, given the growing number of genes of interest linked to drug resistance or sensitivity, we noticed that very few laboratories, as you can see on the chart, very few laboratories restrict the use of NGS to a few routine genes. Indeed, currently, 55% of the laboratories are using panels of more than 50 genes. Even 20% of them are using more than 200 genes in their panel. Dr. Lars, can you comment these evolutions?
Yeah, sure. This is not really astonishing because, you know, since recently, only few targeted therapies were approved associated with some few biomarkers. But recently, with the blooming of the new targeted treatment, and especially also the immunotherapies, the oncologist needs more and more genes. So we clearly see the evolution starting from small panel to middle-sized panel, and now is the time for the big panel. So in order to better decipher the different technologies, actually, as you know, there are two main technologies present on the market, which is Illumina and Thermo Fisher, that are using different ways to perform the sequencing. But also there are different ways to perform the library which is required for the sequencing. So it might be either based on PCR based, like the AmpliSeq technology, or it might be done through hybridization. So we will look into detail what are the different technologies for library preparation and sequencing that are used.
Thank you, Dr. Lars. From the survey, we indeed saw, as you can see on the screen, that the NGS panels, the majority of oncology NGS panels are Thermo Fisher Scientific for 65% of the case, AmpliSeq for 34%, and OncoMINE for 31% of the panel used. So a big majority of them are from Thermo Fisher technology. It's closely followed by Illumina panels, as you can see, the different range of Illumina panels, and then Quiogen and Agilent panels, which seem to be much less used. Bon, pay attention that if you add all the data together, you will exceed 100% because some labs, for sure, are using multiple panels according to their needs. So the amount of biological data is exploding, as is the variety of different data type and bioinformatic platforms. So the technical requirement capabilities of deep interpretation beyond the simple, between brackets, variant calling, and the real support for therapeutic decision for oncologists are now rapidly changing the NGS analysis interpretation platform. So the current dominance of Thermo Fisher Scion Reporter, which is now 27% of market share, as you can see on this chart, is threatened by in-house solution. And also newcomers, you can see Sophia DDM and OncoKDM, for example, which offer deeper and more complete analysis. Well, nevertheless, the traditional platforms are still widely used because if you ask the question to the participant, they mention the automation of this platform for 55% of the participants, the simplicity of the platform, and the low cost of this platform. However, as you will see in a few slides, there are new clinical requirements. And this highlights new weaknesses in the actual platform they have. For sure, the platform associated with some sequencing technologies of the current leaders. So first, our survey clearly identifies some dissatisfaction of the participant with certain aspects. When we ask about the satisfaction rate of the different aspects of the platform, there are three very bad functionalities considered by the participant. What was rated as very bad and poor was the drug recommendation on the actual platform, the bibliographic list associated with the variant font, and the variant classifications. So the worst rated functionalities can also be discriminated by the size of the panel used. If we look more carefully, when we analyze the size of panel used below or above 50 genes, you can see that the drug recommendation of the actual platform is even worse for the large panel users. And the opposite, the bibliographic support for the small panel user is very important. So it seems contradictory, but it makes sense because the small panel user is often linked with approved drugs, very well-known drugs, where the big panel user are dealing with much more complex markers and need an interpretation for the drug to use. So, Dr. Las, can you comment on these new market trends?
Yeah, actually, we clearly see that with the coming of the immunotherapies and the combinations of the different already existing therapies on the markets, actually the oncologist needs more and more different markers like the microsatellite instability, the tumor mutational burden, as well as the expression of some key protein and copy number. And actually, we need to gather all this information in one comprehensive report. So clearly, the needs, the clinical evidence, and the way to gather all this information is becoming more and more a requirement by the oncologist. So it's not astonishing at all.
Thank you. If we look at the survey once again, these trends are really clear. Even if 17% of the users are happy with their actual solution, having a consolidated view on the patient clinical data, on the immunohistochemistry data, and the copy number variation is very important for the participant. They want to have this analysis in the future platform they will acquire. So they need much more than a variant listing, and they want to receive much more comprehensive and interpreted report, including drug, but also clinical trials. So if we have a deeper look on the large panel users, it strengthens the needs, but also adding another needs is the tumor mutational burden analysis. It makes sense with large panels. One participant were even asked to rate this new interpretation platform functionalities in order of importance. It is not shown on this chart, but MSI and TMB data clearly come top. If we now look at the small panel users, so below 50 genes, they are more in need of rich bibliographic support, what we saw at a few slides before, but also variant classification based on their local clinical guidelines. So what is interesting to see is that all these new interpretation needs can be found in the OncoKDM platform. It has a unique feature of indeed answering all these market needs. So OncoDNA is known for oncology market in the oncology diagnostic market since years, but they launched in 2017 OncoKDM platform because we believe that it answers a lot of your needs. Like we added drug recommendation, clinical impact based on multivariant interpretation, analysis of MSI, TMB, immunohistochemistry. We added also information about the clinical data of the patient and CNV analysis and interpretation. So OncoDNA has accumulated also many evidence of the interest of this comprehensive approach. Can you comment on this, Dr. Las?
Yeah, like you mentioned, actually, this is not something which is really new for us. Because since the beginning, we developed our own panel for sequencing that we complement with additional text, because we really believe since the beginning that actually DNA is not all. and that you need also the combination of the analysis of some proteins in order to decipher the complex pathway and provide the oncologists with better advices for their patient. And we made a publication where we demonstrate that really the addition of those immunohistochemistries or those additional tests provide the oncologists with more options for their patient. And recently, we have been really pleased to see in a clinical trial that has been published in ASCO this year that to better understand the benefit of the anti-mTOR for breast cancer, the analysis of a peculiar phosphorylation of a protein was really associated with a benefit for the patient. And actually, we did that several years before, even by analyzing a more complex pathway than just this protein. So together, this information is something that we are doing since the beginning of OncoDNA, and we translate that in OncoKDM. So among the different technologies we can handle, actually the one we are the most advanced is OnCOMINE. Why? Because actually we are using this AmpliSeq technology since the beginning, and our panel are based on these technologies, meaning that to provide our customers with a comprehensive report, we needed to develop original algorithm and original way to analyze the data. And by instance, we developed our own way to perform copy number analysis, which was not possible at that time, meaning that even with hotspot panel or very few applicants, we can perform copy number variation analysis. And in addition, we had to develop our own way to perform the identification of some insertion deletion at a lower frequency that is associated with the variant color associated with the device. So don't be misunderstood. I don't say that the variant color is bad. This is not the case. But we are able to go much further than the variant color which is provided with the device.
Thank you. So we have also a good news for you today also, because it's nice to hear this this information, but we can suggest also all the participants of this webinar to challenge us. So it means that we offer all the participants of the webinar the opportunity to send us a few retrospective OncoMINE samples for interpretation free of charge. So, and then you can see if eventually we found CNV that you may have missed with your technique. So just a small question, if you are able to give us your interest about this solution, we have a small poll that you can answer. If you are interested to test some samples, we will be happy to do it for you. Or eventually, if you have other type of sample to test with retrospective data, we can for sure also help you. Still a few participants to answer, so we will close the poll in a few seconds for the last. Thank you. A few, yes, still a few. Okay. Thank you for that. So we will contact you after the webinar in a few days to go through this offer. So of course, I hope you were interested with this first result of the barometer. We will continue this barometer again and come back to you with maybe other information in a few months. Nevertheless, you will get the report, the printed report, individually after this webinar, so we will send you this report. You can also visit the OncoKDM website, where you will be able to download the folder if you want. We didn't want it to go too much in deeper into OncoKDM today, but you can go deeper by yourself, eventually ask a demo or watch some movie we put on the website. So we are really happy that you were present today. So this webinar is recorded, so you will be able also to show it to colleagues afterwards, or to transfer the link afterwards. Now the floor is yours. If you have some question, we will be happy to answer some of them. Aubrey, I don't know if you still have some question to answer.
So no, thank you for this very interesting session. Thank you, Marc, and thank you, Jean-François, for sharing the outcomes of this comprehensive report. Yes, in fact, this is the time to raise your question by using the internet question box at the right side of your screen. So if you have a question, is the time to raise it up in the question box. Okay, you have some question. I will read it and then give you the floor to answer in a few seconds. Okay, for the first question, have you developed an algorithm to detect CNV? Want to wait. Tu sais mettre ton son?
Okay.
Okay, so the question is that we developed our own algorithm to perform copy number variation. So that might be used on OnCOMINE based panel as well as Illumina based panel. So actually we are agnostic because we suited the parameter according to the different platform. And actually there is a website you can look at, it's OncoCNV, it's free of charge. So you can have a look at different publication, different data. And actually it's also advices by Illumina tech support to use this color for copy number variation.
Okay, thank you, Jean-François. Second question, do you have a knowledge base for variant interpretation? Is it correlation-based like KGen or correlation-based based on artificial intelligence learning like SOFIA?
So we developed our own database, and I will be very pleased if it was artificial intelligence based because I will have less work. However, we noticed that actually you need to read the articles. So we have scientists that are reading the articles and that are writing some conclusion if there is something interesting. And we have developed a validation process where we have other scientists, expert in oncology, that has to read and perhaps confirm or not confirm and validate the conclusion made by the first one. So it's really a human process. So we are browsing available literature and database, and we write the conclusion based on the reading of the article. We don't rely on any abstract title and auto database without checking the information ourselves.
Okay, third question, how does input quality affect the result? I think the question is related to FAP sample, but it doesn't really matter with OncoKDM use.
So actually, regarding the conclusion, actually it affects the data themselves, because if you have a bed fixation, actually you will generate a lot of variants that are related to chemical issues. And in OncoDNA, we develop some filter to remove as much as we can all these artifacts that could lead to false results. But this is something which is related to the original sample, so we have to deal with that.
Right. Next question. Can your platform, so can OncoKDM detect structural variation?
Yes, we can, depending, of course, of the technology used. So we can either detect those based on DNA-seq as well as RNA-seq.
Okay, of course, the question regarding the cost of the analysis, but how much is cost to analyze NGS sample?
Oh, so the question is easy to answer. We have a solution for every kind of clients. So it's really for small panel user, big panel user, the number of samples. So so the simplest way is to ask us which are your needs, and we will come back to you with a quotation, a very good quotation.
Thank you, Mark. Thank you, Jean-François. I think we have answered all the questions. So if there are no more questions, we can now close this session. And we'll be, of course, very happy to welcome you for our next webinar. So stay tuned by following us on Facebook, LinkedIn, and also Twitter. Don't hesitate to visit our website, www.oncokdm.com. And you can also download the OncoKDM brochure or requesting directly a demonstration. Thank you, Mark. Thank you, Jean-François.
Thank you for all the participants. Thank you.
Thank you all.
Bye.
Bye.